Thank you, and thank you to the organizers for inviting me to speak today. And I've been tasked to talk to you about influenza viruses, or to start off the couple speakers talking about influenza. First, the research focus of my lab really focuses on pathogenesis of RNA viruses, especially since going to St. Jude Children's Research Hospital looking in high-risk populations. And as you can see, we do a lot of different aspects, and we work on both flu and astroviruses, which are enteric viruses. So typically, in a talk, this is where I ask people in the audience, how many of you have had influenza virus? But this year, stop, don't put your hands up yet. I think I'm gonna ask a different question, ask how many of you haven't had influenza virus this year? Haha, I bet you have, and you just don't know it, and I'm gonna show you why. This year is been a lot of job security for us in influenza, in that we have widespread disease throughout the country, and it's a very severe year. You can see that we've had a large number of, a lot of morbidity, mortality, we're having packed ERs, and actually IV bag fluids, and it's just continuing to get worse. If you look at, this is from 2 weeks ago, this is influenza-like illness activity levels according to the Centers for Disease Control around the US, and you can see California's in red. If you look at the map now, almost every state is red, and there's that much influenza circulating throughout the US. Now, this is what I'm talking about. We've heard about the typical influenza-like illness, right? Where you have a high fever, body aches, cough, sore throat, you feel really bad for about 2 weeks. We know it can lead to more severe disease, like pneumonia, and God forbid you end up with man flu. That is the worst thing that can happen to you, is to get this. However, I think what most people don't appreciate is that we have a lot of people that actually get flu and have mild disease or completely asymptomatic. We've seen this in our human surveillance, where we've had to change the definition in order to capture influenza. And I finally found a paper from 2014 where they actually went through and looked at this and found that 77% of people that have flu would not be considered with flu-like illness because it's either so mild or asymptomatic. That's why I think our numbers this year of influenza cases, and probably every year, are much higher than what we could expect or predict. We're not alone in the world. This is from the World Health Organization, and you can see influenza is circulating throughout the world. Here in the US, we've got about 21 to 30% at the time of this map of people with influenza-like illness having flu, but you can see in many countries it's actually much higher. And there are 3 different strains of flu currently circulating across the world. And the problem being that, I'll show you this. Here in the US, we have a strain that's particularly nasty, and I'll tell you why that is. Now, I am part of the World Health Organization network. We actually have a collaborating center, and we knew from watching, or predicted from watching what happened in the Southern Hemisphere in the summer, that it was going to be a bad flu year. And here are 2 graphs that I generated using WHO data from FluNet that shows you what happened in Australia. And they had the worst flu outbreak in record. And this is a percent of people that were positive of samples, and this is a number of influenza-like illness. So the red graph is what happened in 2017, and the number of people or samples that were flu positive. I think what's really telling is if you look at these light gray sort of lines, that's what happened during the 2009 pandemic. And you think there's always much more flu during a pandemic. Not this particular year in Australia. So, as I mentioned, the other problem is it's an H3 year. What does that mean? Well, H3s are like the bad boys of the human influenza seasonal strains. Why is that? One problem we have with them is that they like to go through antigenic drift. And these viruses drift a lot. And we're going to hear more about that from Dr. Jude in a couple minutes. The other problem is they are bugaboos to make, and that's a real scientific word, to make vaccines to. The reason for that is we make our flu vaccines, at least here in the US and many parts of the world, in embryonated chicken eggs. With these H3 viruses, people don't appreciate that even if we are spot on with our prediction of what strain we should use for the vaccine, what we can't predict is what's going to happen when we passage them through an egg. And what typically happens is they change glycosylation sites right in the antigenic head of the hemagglutinin, meaning that your vaccine strain is antigenically less effective than the cellular strains that are actually circulating through people. This is a real problem. Australia is moving towards using cell-based vaccines. The other problem with the H3 viruses is they will not grow well in cell culture. So if you can't grow them, you can't isolate them, you can't grow them up to do anything with them. They also don't hemagglutinate red blood cells. And for good or bad, that is still our gold standard for quantitating flu, and mostly bad, as I see John making the same face I make when we think about HAs. And finally, original antigenic sin, or immune history, plays a big part in how you respond to flu. And most people have been imprinted as a child with H1 strains, not with H3. So again, this leads to problems when you see an H3 virus. And Helen Branswell actually wrote a really nice article in Scientific American about what's wrong this year and the problems with H3 vaccines. I would encourage you to read to better appreciate what we have to deal with influenza during an H3 year. So I don't know if anybody else finds this as ironic as I do, that one of the worst flu outbreaks, we can't call it an epidemic yet, actually falls on the 100-year anniversary of what was the worst recorded influenza pandemic in history, and that was the 1918 flu. And the predictions are anywhere from 20 to 50 million people died from the 1918 flu. Of course, when we're writing grants, it becomes hundreds of millions of people, and of course it's going to happen again. But it really was a very intriguing virus. You can see the death rate in October, and it's the only time in recorded history that we actually saw a decrease in the life expectancy rate. And I love this chart showing even in 1918, where we couldn't jump on a plane, looking at how fast that virus spread through the US. One thing that this season has told us, and it's probably the only time I've ever agreed with Mike Osterholm about anything, but what he said is that we're not ready for a flu pandemic, and that's probably true. So what are we going to do? How are we going to fight this constantly evolving virus, right? It's like hitting a moving target. Well, the first thing we have to think about is we have to understand what's out there, what's circulating in the environment. And we have great networks set up to actually monitor this. Through the World Health Organization, there's a global influenza surveillance network looking for flu in people. It's comprised of 6 collaborating centers. We at St. Jude are one. This is our official title. I'm not going to say it because it's an absolute mouthful. Along with Centers for Disease Control, London, Australia, Japan, and China. And we are responsible for coordinating influenza surveillance in 144 different influenza centers throughout the world. And what they've done is set up different kinds of networks like severe acute respiratory illness, ILI, working with physicians and office visits, and public health laboratories to understand what viruses are actually out there and what the prevalence is. However, we know with flu, we can't just focus on humans. Now, this is the wheel of influenza misfortune. And what you see is a duck in the middle, because as we know, waterfowl are the proposed reservoir for influenza viruses. And these birds typically get flu. They don't have any clinical symptoms, but they will continue to shed virus as they migrate. So unlike people where it comes out respiratory, this comes out as poop. Gets into the environment, can directly pass into sea animals, horses, poultry, pigs. And notice there's no line to people, because we never thought those wild bird viruses could get directly through people. We thought they had to go through an intermediate, and that was pigs. But we now know that's not true. And bird viruses can get directly into people with devastating consequences. This is really a cat with a biosafety mask on it, because we've seen that these viruses can get into cats, and then they can transmit cat to cat. They can get it from people. And we've got a lot of things going on in the dog world as well. So to really look for flu, you also have to have groups that are looking outside of just humans. And that's where NIAID, starting in 2007, funded this network. It's the Centers of Excellence for Influenza Research and Surveillance, or CEIRS. You can see there's 5 of these, with us being at St. Jude. And again, our mission is to look for influenza and then understand it. And at St. Jude, we're somewhat unique in that we really focus on influenza at the animal-human interface. And this shows you all the different collaborators we have around the world where we're actually looking for flu, either in birds, in swine, in humans. And because we have such a great network set up, we've also been tasked with looking for things like MERS and Zika. And what we do, the joke is, if anything, if we can catch it, whether it's flying, running, or swimming, if we can catch it, we're going to swab it. And that's exactly what we do. And then we use those viruses to actually understand impact on the immune response, disease pathogenesis, and understand if these viruses are transmissible. And we coordinate all this information with other government industries, and we bring it to WHO, and we're responsible for deciding vaccines to emerging zoonotic viruses, and then we actually have to make the vaccines many times at St. Jude. We've also, I think the most important thing is we're looking at the animal-human interface, because that's where the action's going to happen. And it can be in many different kinds of people, but particularly in high-risk populations. So how do you know? So we isolate a lot of viruses and identify a lot of viruses every year. How do you know if that virus that you've isolated is a risk to people? Well, we have a viral characterization pipeline that really focuses on risk to humans, and we can literally sort of categorize these. If we see any viruses like this, we consider them a higher risk to humans, or we can look to specific groups, whether it be poultry or swine, and we look for things like molecular determinants of virulence, receptor binding specificity, growth in primary cells, population-wide immunity, and then we can feed it into algorithms either from the CDC or WHO to give these a quantitation and say, are these a possible risk to humans? And honestly, I can tell you the viruses that we have circulating in swine right now are probably some of the highest risk in their potential to cross into humans, because they've already made that jump from birds into mammals, are now circulating in swine, and we're seeing them transmit to humans, especially kids, at state fairs every year. And this is just an example of some of the vaccines that we've made at St. Jude for the WHO network that got put into government repositories as seed stocks in case these vaccines are ever needed. So why is animal surveillance important? There's a lot of questions about why are you doing this? Why are you looking in wild birds? Who cares? Well, we have to understand what viruses are in the gene pool, what genes are out there, which ones can actually spread to and within mammals, what's the risk to humans. It gives us a lot of information about basic virology in terms of reassortant and antigenic drift, and we desperately need them for things like diagnostics and therapies. Even if we make a universal flu vaccine, you still have to understand what's circulating in nature to ensure that that vaccine is going to protect you. And the goal is to go from swab to sequence to public health. Now, this has been difficult to do for the past 3 years because we've been under a moratorium. That has been raised, so now we can get back to business. And we know the other problem is that a lot of the funds are being redirected from surveillance to making universal vaccines. So, is it possible to make a universal vaccine? I don't know, maybe, but it's definitely possible to improve our vaccines. Easiest thing right out of the bat, as far as I'm concerned, is get it out of the eggs. Put it in cells. That's what Australia is doing. We'll see how that improves their vaccine efficacy. We need more therapeutics, and we're going to hear more about that from Andy Melly next. We need better cells. Right now, most of influenza viruses are grown in canine kidney cells. Really? I mean, that makes absolutely no sense. For swine viruses, it's even better. It's swine testicle cells. I don't know what that has to do with the respiratory, but that's something very practical and sort of unsexy science that we're doing is we've developed primary respiratory nasal, tracheobronchial, and alveolar epithelial cells from a variety of species including humans. And we've made cell lines, and we've shown that you can isolate more viruses. They grow to higher titers. Genomic stability is in place because we're not changing species as we passage. And somebody has got to make us better assays than a hemagglutination inhibition if we really want to understand flu. And finally, we have to understand disease severity. What are the molecular determinants within the virus? What are the cellular factors? We've worked a lot with a specific integrin we've shown is very important and a good target for making improved vaccines. We need to know host genetics, signatures of more severe, less severe disease, immune correlates of protection, and how does immune history play into this? The big thing is we need to start thinking outside of the box, right? There's so much dogma in influenza, and part of the problem is it's really not a big field. And most of the people that have been trained in flu, there are some of us that are excluded, trained in one of probably, 4 laboratories, and they haven't fallen far from the tree. So I think we need to start thinking outside of that, throw all the dogma away, and start moving forward. And one thing that my lab's been doing is really trying to understand this disease severity, and it's blossomed into a lot more. As I mentioned, we're looking at this integrin, but what we're specifically interested in is this, is how does influenza or obesity impact influenza virus? Now, this sort of came to us during the 2009 pandemic, where epidemiological evidence suggested that obesity was associated with more severe disease. Now, if this is true, this is a huge problem, haha, pun intended, because you think about flu causing pandemics, obesity has really become an epidemic. This is from the World Health Organization in 2014. It's the number of men aged 18 or older that are considered obese. And it's in red, which you can see, and it's increased even more. Women, unfortunately, are even worse. And the WHO has predicted if we continue on this trend, by 2021, 3/4 of the world is going to be overweight or obese. We're not having problems as much with malnutrition, it really is obesity. And the group that's becoming fattest, fastest, are children, are toddlers. If you look at 4-year-olds, 75% of them, especially where I live, are actually obese, not even overweight. We're also making our animals fat, literally. So if you think about obesity, it's really straightforward, right? You burn more calories than you consume, you're considered average or healthy weight. Changes if you consume more than you burn. Now, it's all based on body mass index or BMI, which is a problem because all that takes into account is height versus weight. And that can be very deceiving, but it's the best that we have. But obesity is actually a big problem in that it causes an immunocompromised state because its adipose tissue is actually an endocrine organ secreting a lot of pro-inflammatory cytokines, which can impact your immune cells. So we asked, does obesity truly impact influenza virus? And we decided to use 2 models, diet-induced obesity or OB-OB, which are the genetically obese, because of course, nobody is overweight because they eat too much. It's always because of genetics. So we wanted to model both. This is Albert. He is the fattest mouse we've ever had. He went 80 grams. So he's a big boy. But you know, they're happy. They don't try and bite you. They don't try and run away from you. They just sit in the cage and eat. You obviously do not pick them up by their tails because nothing good comes of that. But we took these obese animals and we asked if we give them flu, whether it was the pandemic strain or any influenza virus, what happens? And what we can see is regardless of the type of obesity, we have increased morbidity and mortality. And you know, mice are not normal hosts for flu. We typically have to adapt viruses. But for obese mice, there is no adaptation required. I can take the virus that probably shouldn't be in me anymore, but was in me last week, and put it in a mouse, and I can guarantee you that mouse is going to get sick. One of the big problems is the virus spreads much deeper in the lungs of these animals. And this is using Andy's beautiful reporter virus. Here's wild-type mice with his luciferase virus, and here's the obese mice. And you can see how it's spread throughout the respiratory tract. Now, one of the nice things about using a reporter virus is that you can monitor mice in real time without having to euthanize them, right? Not true with the obese mice. We have to take off their coats because they have so much adipose tissue around their lungs. You literally can't image them without opening them up. So rather than give you a lot of data, because I don't have a lot of time, what we found is that obesity is a huge problem in that we get increased viral spread and titers. Animals end up with acute lung injury and acute respiratory distress syndrome. There's problems clearing the virus, and it really is a susceptibility in that our mouse lethal dose 50s go from maybe 10 to the 6 to 10 to the 2. There's decreased wound repair and exposed basement membrane, and what that means is that there's an increased chance of developing secondary bacterial infections. And what we see is that's absolutely true, is if we do a co-infection model where we administer bacteria 7 days post-flu, even in a lean mouse where we'd have no morbidity, our obese mice will die. And it's because the viral titers and the bacterial and viral titers are much higher, and the lung injury is more severe. And again, you can see this spread issue with both the virus and the bacteria. So one thing about these secondary bacterial infections is that you can protect lean animals with vaccination, whether it's to the virus or the bacteria. That is not true in the obese animals. Whether you give them flu vaccine, Pneumovax, Prevnar, even vaccines under development to pneumococcus, you can't protect the obese mice. Antibiotics still work, but they're not as effective. And this vaccination issue in the obese animals is not limited to the secondary bacterial infection. It is true just during primary infection. So if you look at hemagglutination inhibition titers, which are how many antibodies do you have, what you can see is there's an inverse correlation between having good titers and weight. The heavier the weight of the mice, the worse they respond to vaccination, and we can't protect them. So we asked, what if we give them an adjuvant? And we've done that. Here's lean with alum or squalene or obese mice. We can boost these antibody titers, whether they're hemagglutination inhibition, microneutralization, which is really the gold standard you want, stalk antibodies to this magical universal vaccine, or even antibodies to neuraminidase. If you give an adjuvant, we get into protective titers, according to the dogma. But you still can't protect these mice from challenge at all. Here's your lean mice, where we've got PBS, you've got just the vaccine alone, and the vaccine plus any kind of adjuvant, complete protection. Nothing in the obese mice. And it's because we're not decreasing viral titers. Absolutely, it doesn't matter if it's in the lungs, if it's in the nasal passages. Blue is your lean mice, green are obese, and what you can see is days post, you start to get better protection, but we are not clearing the virus from these animals. So we asked, what happens if we increase the amount of vaccine antigen we give them? That's what we have to do in aged adults now, right? And we did that. We used base, or we used high vaccine, plus or minus adjuvants. Again, we increase these supposed immune correlates of protection, still can't protect the animals. We prolong the agony, but we don't get protection. Even if we lower the viral challenge, we have to get to basically 1 MLD50 or 1 viral particle to protect them. So there's a real problem here. And we know it's a twofold problem, because if we take sera from lean animals and passively immunize obese, we know those antibodies work just fine, can't protect them. So there's something wrong with the microenvironment. But if we take obese serum and put it in lean animals, we can't protect them either. So there's something wrong with the antibodies themselves. And what we know from looking at arrays to HA is that both the breadth and the magnitude of the vaccine response in these obese animals is impacted. It's decreased, and we're recognizing different epitopes on the hemagglutinin than we do in a lean animal. Does this happen in people? Because we save a lot of mice each year from flu. I mean, we are really, really good at it. But does this actually transmit or translate to humans? And the answer is maybe. We've been doing studies with Melinda Beck at University of North Carolina, where we vaccinated different weight people, and then we look at vaccine responses over time. And what I can tell you is overweight, obese, and we've gone up to BMIs of 40, which is morbidly obese. 1 month, 3 month post-vaccination, it's no different than a healthy weight person. But when you get to 6 months and a year, those obese, overweight obese people have lost their antibodies much faster. And it does correlate with age. One thing we found this year is that in spite of vaccination, even if it's an H1 year, those obese people are still more likely to get infected with flu. Okay, so big problem, lots of people to study. We've got increased susceptibility and disease severity, decreased vaccine responses, prolonged viral shed, and that's in quotes because in mice they don't really shed virus, but what you can see is that there's a clearance problem, and we actually see this, or we think we see this in preliminary studies in humans where overweight, obese, and this is from kids, are shedding virus longer. We're hypothesizing this is actually changing the viral population, and this may be a reservoir for this antigenic drift that we're seeing. But we don't know how it really impacts the virus in transmission because you can't do that in a mouse. So what we did is we decided to make overweight ferrets. Ferrets are the gold standard for influenza, especially transmission and vaccines. So what we did is we put them on ferret Happy Meals, right? So high-fat diet, and then we monitor weight over time, and you can see they like to eat, they gain weight. They are different. What we notice is that they actually get squishy. That's what my people told me. I said, you have got to quantitate squishy for me, because we're not putting that in a paper. So luckily, I have a graduate student who has a background in exercise physiology. So what they were able to do is measure the circumference of animals when they're anesthetized, and we got calipers. So now we can actually measure squishy. So rather than being lean and muscular, these animals develop visceral fat like people do, good or bad. The other thing we found out is if we treat these animals, we give them, rather than just their Happy Meal, they get a can of Coke. So if they get sucrose water, that will also increase the weight. The problem with that is these guys are like crackheads when it comes to sucrose water because they will literally lay under that water and drink it until it's gone. And we ran into problems with them not eating because they just wanted to drink the sucrose water, so we had to stop that. But what we now have, or we hope, is this model of diet-induced obesity in a ferret. So what happens? Luckily for us, it looks just like the mouse model, in that if you give them influenza virus, so most strains of influenza in ferrets, even if it transmits beautifully like the pandemic virus or what's circulating now, they really don't get clinical symptoms of disease. The obese ones do. What we see is they lose weight, they develop fevers, and their overall clinical scores, they don't want to run around, they're really sick, are higher. We always see sneezing, but now we've got coughing, we've got eye and ear infections, which we've never seen in the ferrets before, and we're having problems with secondary bacterial infections, just from things in the environment. We still see, if you look at nasal wash titers, days post-infection, that these ferrets are shedding much longer. We're seeing shed up to day 21 in the obese animals. And if you look in the lungs themselves at titers, what we can see is the viral titers are much, much higher. So this is log scale, lean and obese, upper left, upper right, mid right, lower right. Lean animal, you can see the virus really doesn't get down deep in the lungs, and that is absolutely not true in the obese animals. But what about transmission, right? This is the million-dollar question with flu. What we decided to do, and we did this with Elodie Gedden at New York University, is we gave ferrets a strain of flu that typically does not transmit in the ferret model. We know this, other people know this. H9N2, we directly inoculate ferrets, either obese or lean, then we put ferrets in direct contact, so they go in the same cage, or an aerosol contact. So they're separated by basically a mesh grate. They can't touch each other. There's no way they can contact each other. And we asked, do we see transmission? This is nasal wash titers again, days post-infection. And that's from the directly inoculated animal. Black and gray is our lean animals. And what you can see is our directly inoculated animals got infected. And there was transmission by direct contact in 1 of 3 animals. All right, that's good. What happens in the obese ferrets? Everybody, all the directly inoculated get infected. They have slightly higher titers. They shed it much longer. In this case, both the direct contact and the aerosol contact, 100% of them got infected. So something has changed. Either those animals are more susceptible or the virus has changed. And it's probably both. But with Elodie, we looked at the viral population. And this is from the lean animals. And it's just showing you the percent of the population within different regions. And this is just HA. We've looked throughout the genome, have changed. And what you can see is we have enhanced viral populations, or an increase in the viral population with more changes in these obese animals. So what we're now asking is it this disease susceptibility, or are these guys actually going to end up being super spreaders? Okay. And will this go beyond influenza? And the answer is going to be yes. So with that, I have to thank everybody in the lab, everybody that works with us, because these are big studies that take a lot of people, and remind those of you that are virologists to go to ASV this year, because it's going to be fantastic. And that is it. Thank you.